All right, let's go ahead and get started with our first afternoon session, Human Dimensions. Hopefully everybody's digesting their food, got some more energy. So we're starting off this session, the standalone session, was with David Trimbach with Washington Department of Fish and Wildlife. And the title of his talk is going to be Place of Salmon, highlighting the relationships among salmon, people, and place in Washington, and the potential for social science contributions to salmon restoration. So, David, take it away.
Good afternoon, everybody. Thanks for coming back. Thanks for coming back and listening to me talk about human dimensions, of all things. My name is David Trimbach, and I'm the conservation social scientist at the Washington Department of Fish and Wildlife. Yes, that is a position, that is a thing at the agency. It is fairly new. I've been at the agency for about a year and a half, and it's not just me. I get to work with staff across the agency, across the state on all sorts of projects. I get to work with external partners, again, on all sorts of projects. And I also work as part of a team. So it's not just me. We have a natural resource economist. We have a diversity, equity, and inclusion data analyst who also does geospatial data analysis. And then we also have an environmental justice coordinator who uses social science and data to inform that work at our agency. And I was invited, so thank you very much for the invitation, to talk about human dimensions, which means many different things to many different people. And I will illustrate that in my presentation, which focuses on place of salmon, highlighting the relationships among salmon, people, and place. Who knew that was important to Washingtonians and to those in this room? And the potential for social science contributions to salmon restoration. And so this presentation includes two different parts. One is kind of a crash course on what are the conservation social sciences? Like, what does that even mean? What does that even look like? Hopefully, it's interesting to you all, and you can see the potential contributions to your work. And then I'm going to show an example from a project I've been working on for a few years now, focused on sense of place in salmon. So people's connections and identities associated with salmon, and why does that matter? And that project is funded by the Puget Sound Institute of the University of Washington. And while it does have a heavy Puget Sound focus, which is where I used to conduct almost all of my research, we did incorporate data to reflect these relationships across the entire state of Washington. So what does this even mean? What does this look like? What are the conservation social sciences? As you can see on this wheel here, we have this diagram of the conservation social sciences. These are, as defined by scholars, these are a set of scientific disciplines. These are sciences. And there are many of them. And they are not all the same. But they do share this focus on the conservation-relevant aspects of human societies. Or if you think about that a little bit differently, think about it as the human elements or human dimensions of conservation and natural resource management. So it is often framed as a topic of study, a subject of study. And if you look closely, for those of you maybe with glasses, you can also see human dimensions on the wheel itself. So in some contexts, in some universities, it's also its own interdisciplinary field of science. And there actually aren't that many institutions that actually have a human dimensions department. Colorado State University has the department of human dimensions. They kind of have a monopoly on that term in that field as a social science. And that kind of cobbles together information from all sorts of social sciences to create that department. And then the conservation social sciences are often conflated with, but they are different and they do contribute to other things that are very human in our world, including communications, education, outreach, social marketing, a very popular thing right now, and human dimensions as a practice. And so many natural resource departments, especially at state agencies, have human dimensions specialists who kind of end up in these positions which are kind of like the catch-all of all things human. We don't necessarily, it's not communications exactly, but it's these other things. And so you see a lot of diversity when it comes to what human dimensions looks like in conservation and natural resource management. So it's not necessarily one thing to all people. And when you look at the conservation social sciences specifically, you can see a lot of variety. And you see this list here. And so most conservation social sciences are actually subdisciplines of larger social sciences. So larger social sciences like sociology, economics, political science, geography, so the conservation social sciences are all kind of like a part of that. So I'm a geographer by training, but more specifically, I'm a human geographer by training, which is a conservation social science. So we study the relationships, interactions between people and the natural world. What do we study? We study anything social. If it's social, we probably study it, and we've probably been studying it for decades. So using theories, methods, different forms of data analysis, we study things like governance, decision-making, values, behavior. Again, if it's social, we study it. And how do we do research? Just like any other science, we use methods. We use tools for data collection and analysis. And not all social scientists are trained in the same tools. Some specific social sciences kind of specialize in specific tools, like anthropology. They really have the monopoly on ethnography. That's really where that comes from, which is a deep, qualitative, long-term tool to understand the social world. And it's important to recognize that not all tools are the same, nor are they always appropriate to answer every question. So we don't necessarily always need a survey, even though we may really want to do that survey. And as someone at WDFW, that's probably the question that I get the most. Can we do a survey on this? And sometimes we don't necessarily need to do that. And given that this conference partly focuses on scale, scale also matters to social scientists. We also think about scale. We incorporate scale into how we design projects and implement them. And so, as you can see from this image, we might look at different units of analysis or different topics at different scales or multiple scales simultaneously. As a geographer who tends to look at the world through space, place, and scale, it's always baked into my work. Another way to think about scale is when we think about the work that we do using a social ecological systems framework. Traditionally, social scientists primarily focused on the social system or society in their work, while traditionally natural scientists tended to focus on ecosystems or the ecological system in their work. However, more and more scholars and practitioners alike are looking at their work within the context of these two constantly interacting, constantly changing, mutually constitutive systems that are always at play with one another. We can't ignore these things as siloed entities, as siloed systems. They're always interacting. And you can look at that system at various scales, whether it's an individual human interacting with an individual species, or populations of humans in populations of species interacting. And how have the conservation social sciences approached or researched salmon restoration? Have they done this? Yes, they have. And I feel very lucky and privileged to be in the position that I'm in at the agency, partly because I get to work with a really great community of conservation social scientists in the Pacific Northwest. And this is probably the best place in the country, if not the world, to be in to do this work. Because we have amazing ecosystems and social ecological systems with so many questions. And people really value social science in this region. And so I'm just going to highlight some topics that people have focused on in Washington. So some scholars have focused on indigenous food sovereignty, well-being, and fisheries management and their importance. Others have looked at how human values and sense of place informs conflicts within salmon restoration. So people have competing values, people have competing connections to place, and those are often demonstrated in restoration through conflict. Others have looked at how knowledge pluralism is really integral to salmon restoration. We can't just use one type of science or one type of knowledge. We need other types of knowledge to also create effective strategies and outcomes in conservation. Others have looked at the use of community science, often framed as citizen science, and how that can foster equity and justice within salmon restoration, particularly in Seattle. So why do this? Why integrate the conservation social sciences in your work? There are many reasons, and I had a a 5-year postdoc and another position at Oregon State University prior to coming to WDFW, and it was really focused on social science integration into ecosystem recovery in Puget Sound. And so I've had to read a lot about why do we do this? And there are many reasons, and so I'm going to highlight a few. So one is improve the credibility, defensibility, and social acceptability of management decisions. How the conservation of social sciences can help facilitate more socially equitable and just processes and outcomes from the work that we do. It can help us understand why this community over here really opposes what we're doing versus this community over here, which really supports what we're doing. Why are there differences? It can also help frame communication and outreach strategies if our goal is to change people's attitudes and behaviors related to conservation or restoration. And I feel like much of my work seems to directly kind of feed into communications, education, and outreach strategies developed by the agency. And I think overall, the integration of the social sciences, in addition to other forms of knowledge and other inputs that go into conservation and restoration, addresses this need for pluralism, because conservation does not look the same, nor does it require the same inputs in all contexts. It's not a one-size-fits-all problem. And so instead of working on all of our own kind of puzzles, of which is conservation, we should be putting all of our pieces together. And so now that you're bought in, now that you're really interested in this, when can you actually do this? And there's actually been research on when can we integrate social science into what we do. And there was a conference last year where we actually workshopped all of these different things. And so you can actually do it anytime, as this image kind of illustrates. You can do it at the beginning where you're kind of defining the problem, defining your project team, and you can do it at the end when you want to evaluate what you did. Was it successful? You can use social science tools to do that. And given that this inherently means that it's collaborative, the earlier the better. So contact those people who do this kind of work, integrate them into your team. And you can do this in different ways. Create an inclusive budget and team. This compensate people for their contributions. Be a champion, be an advocate. My position wasn't just created overnight. I think there was a decade or more of people, internal and external to the agency, advocating for somebody to think about the social questions when it comes to the work that we do. And if you do end up working with a social scientist, don't do things in silos. Oftentimes we get in silos, we speak specific languages, we use specific terminology, we use specific tools, and then when we come together, it's really hard to communicate. And so when co-creating knowledge, when co-creating projects, when co-creating questions, we get to learn from each other. We get to share values, we get to share objectives, we get to share language as to how we talk about this stuff. And we're here, we exist. Not just at DFW, but the Puget Sound Partnership actually just hired their first internal social scientist to focus on human dimensions in Puget Sound. Many different higher education institutions in the state also have conservation social scientists who do really awesome work. And then federal agencies have been doing this for decades. They've really been the leader in human dimensions and social science. So now that we're all social scientists, I thought that we could then dive into a project as an example from my own work. And this project focuses on salmon, people, and place in Washington. And I'm not alone. This is a highly collaborative project with lots of input from DFW staff and external partners. And so salmon, people, and place in Washington, who knew that that would be a thing, right? Who knew that those things were connected or important? And I think it's important to recognize that salmon have been integral to communities and the region's social ecological system since time immemorial. So prior to white settler colonialism, people here, indigenous people, have had relationships and interactions with salmon long before white settler colonialism. And those relationships are still really important. And there are other relationships that other residents in the state also have with salmon. And they are slightly different and distinct. And this is manifested in lots of different ways. This is reflected in art, of which, here is an example. I think the state of Washington has lots of various examples of art related to salmon. I think you can't go to any town in the state without seeing salmon represented in some way on the physical landscape. And it's also represented in other ways. And so these are just some examples. So one is our most recent poet laureate wrote an amazing book, a poetry book on salmon and why they're important to Washingtonians. The governor frequently uses salmon and orca specifically to evoke emotion among the population when talking about policy decisions or when making speeches. And salmon and orca in particular are often used and framed around our identities, our shared connection to place. Timothy Egan, who is a really famous environmental writer, wrote a fantastic book, obviously a long time ago. And there's a quote from that book that's used innumerable times in a lot of different contexts, which states that the Pacific Northwest is simply this, wherever the salmon can get to. And all of this feeds into and kind of is represented by this debate among environmental historians in the Pacific Northwest about 2 decades ago around this question of whether or not we can claim to be the place of salmon. And so there were historians that debated, yes, absolutely. And there were other historians that said, no, we can't. And so all of this kind of fits into the context of this research. And so this project focuses on salmon people in place with this emphasis on sense of place. And so while there are a lot of anecdotal information, rhetoric, and stories about salmon and their importance to Washingtonians, there's not a lot of social scientific research looking at those connections. And so we developed a project aimed at enhancing knowledge of the relationships among salmon people in place in Washington, which includes specific components and other objectives like how does salmon contribute to people's connections to Washington, or in our case, Puget Sound? And are salmon representative symbols of this place? If the governor's frequently saying and using salmon as a symbol of Washington, would that even resonate with people? It might with the people in this room, but would it resonate with other Washingtonians? So is this the place of salmon? Is this salmon country, as I saw earlier on a pamphlet, which I changed my presentation to reflect? And what might that mean for salmon restoration? So this study focuses on these concepts which are frequently used in the social sciences, sense of place and placemaking, often in environmental psychology and human geography, which is where most of my research sits. So sense of place, if you're unfamiliar, are essentially the relationships between people and place. And those include specific components that we often measure quantitatively. So that includes place attachment, which is people's connections to place, of which we have many, and they're often complex. And within that, there's place identity, and we have many place-based identities. For example, I'm a Tacoman, I'm a Washingtonian, I'm a Pacific Northwesterner, I'm an Ohioan. I hope you don't use that against me. And then we also have place dependencies. So we, as humans, we rely on place for many different reasons. Whether it's for work, whether it's for food, whether it's for recreation, we all depend on place for a lot of different reasons. And those are often qualified and informed by place meanings, which are typically measured more qualitatively. And place meanings are also diverse and complex. For example, if you think about Washington, something typically comes to mind, right? It could be rivers, it could be the mountains, it could be recreating, it could be home, it could be your family, it could be a specific place in Washington. Those are all place meanings, and those inform who we are and how we think about place and how we act in place. And so for this particular study, we actually used a US Forest Service framework to look at place meanings. Specifically, inherent place meanings, which are those kind of meanings that are very observable in the landscape, like physical features. We also looked at identities, and we looked at instrumental meanings, so use-oriented meanings. And next we have placemaking, which is directly related to it, because sense of place typically feeds into making place. So placemaking is literally how it sounds, making place, placemaking. We as human beings constantly make place through all of the various actions we take on the landscape. We are constantly changing places in various ways. As restorationists, we are restoring place. We are engaging in behaviors that are physically moving the Earth's surface in lots of different ways, which make place, which informs how people think and interact with place, who aren't necessarily involved in those behaviors. Traditionally, placemaking research has focused more on the human side, but nature and natural processes like earthquakes, volcanic eruptions, landslides, rivers shifting, and species existing in a particular place also contribute to placemaking. They also help make place as multi-species communities that we exist in. And so this project is looking at sense of place, placemaking, and salmon. How does salmon fit into that? And so this was part of a much larger geographic literacy study, of which we already have a paper out if you're interested, looking at how Puget Sound residents know or think about estuaries. And so this also builds upon previous research that I've done on sense of place in Puget Sound, including on marine shorelines and orca. And then, as I mentioned, this focuses on salmon-inspired placemaking. And so we looked at salmon presence, place naming, so place names associated with salmon, which is often reflecting sense of place or the historic or current ranges of species, and place public art funded by the state, and place-based actions like restoration. So we're looking at how salmon is reflected on the social ecological landscape of Washington. And so we did this by conducting a survey in Puget Sound. 12 county residents, 407 respondents participated, and the survey results were generalizable by age, sex, and county of residence. And we also collected a myriad of geographic data aiming to understand and visualize salmon's imprint on the social ecological landscape of Washington. And so why do we do this? I could talk about sense of place all day, and I've taught classes on it, but why do we focus on this topic? There's many different reasons. One, sense of place is used as a measurable indicator of ecosystem health and recovery and human well-being as it relates to the natural environment. And there's actually multiple indices used in the Puget Sound region by different entities, which include sense of place as an indicator, including the Puget Sound Partnership. It's also used to gauge responses to environmental change and restoration actions. It's actually a common practice in parts of the United Kingdom to do a sense of place analysis of communities prior to infrastructure installation or removal, because large-scale infrastructure is going to change the landscape, and that's going to impact people and how they feel and think about place. It also is a motivator for pro-environmental behaviors, and it's also related to environmental activities, like shellfish harvesting, hunting, fishing. People who engage in those activities, even restoration, are building a sense of place through those actions. So I'm gonna ignore this because this is focusing on sense of place, people, salmon, remember that? So what are the results? So overall, we found that, in general, people in Puget Sound have a really strong sense of place of that place. And this is validated by other studies. We actually use the same questions from other instruments. And so this is really validating, especially for me, because I've done many of these studies. So people have a really strong connection to place. And they also have different place meanings associated with Puget Sound, notably identity expressive meanings. So those are meanings associated with individual experiences, people's emotions, maybe familial memories. And so people are connected to place. How about salmon? Where does salmon fit in? We actually found that salmon does contribute to how people connect to place in different ways, with the exception of dependence. So people are, in the Puget Sound at least, not depending as much on salmon to meet their needs or to meet specific goals, with maybe the exception of recreation. But in general, people do see salmon as a contributor to how they feel and connect to place. And this was also reflected qualitatively in the various meanings that they shared, with the most prominent at the bottom being instrumental, so use-oriented meanings. People do still very much so associate salmon with recreation, with food. And we also asked whether or not people saw salmon as a symbol of the region at different geographic scales. And overwhelmingly, those who participated said yes. Salmon does represent Puget Sound, it does represent Washington, it does represent the Pacific Northwest. It is this thing that evokes some shared sense of a community and place. And we also asked, how much does salmon contribute? Because many things obviously contribute to how people connect with place. So we created a scale from 0 through 10, asking how much does it contribute, and overall most respondents were 7 and above, which was way more than we expected. And then we also collected a whole bunch of geographic data, and I had my geospatial analyst work on this. And so you're going to be looking at a series of maps quite quickly that look at salmon on the landscape, both ecological and social data. So this is salmon and steelhead distributions, both observed and estimated. These are salmon habitat restoration projects, so human beings engaging in activities that are modifying the landscape related to salmon. This might include work that you're associated with. Fish passage barriers, thinking conversely, other types of activities that we engage in on the landscape that may impact salmon. Corrected fish passage barriers. Aquaculture and hatcheries that we construct on the landscape. Public roads, place names, public art funded by the state, and public schools, all named after salmon, reflecting senses of place and reflecting historic or current ranges of the species. Business names. Businesses really like to use native species in their naming and logos, as someone who's done studies on that before. And then this is all of that information together, which we're calling salmon instances on the landscape. So looking at all of those things together, right? So as you can see, there's a lot in Western Washington, but also not just Western Washington. This is reflected across the state in a myriad of ways, illustrating, I think, to a certain extent that this is the place of salmon. So overall, I have 1 minute. Salmon are important to people. You all know that. They are also important to residents in Puget Sound and the state, and it's reflected both in our survey results and in our geographic data that we collected, which I think partly demonstrates the role of salmon in people's well-being, people-place relationships, and potential reactions to salmon restoration or lack thereof. Salmon matter to people beyond recreation and economics. I also think it helps validate recovery efforts, all of the work that people in this room do. So some next steps, our work is already informing communications, outreach, and education. We already have a pilot test at a school district in Puget Sound where they are actually using some of our sociocultural maps in classrooms to talk about why salmon matter to people. So in conclusion, think about sense of place in your work, and also think about the social sciences and what you do. Thank you.
Sorry, we don't have time for questions. We've got to keep moving along, but there's break time after to ask David and kind of ask more questions about salmon in place. All right, so our next session is species survival and life history. And our first speaker for that session is Josh Williams at WDFW. And the presentation title is Life History Characteristics of Bull Trout in the Wenatchee Basin.
Yeah, thanks for that last talk. It's always an important sort of realization to keep in mind the human component. So yeah, cool stuff. And yeah, thank you everybody for being here. My name is Josh Williams, and I'm a fish biologist with Washington Department of Fish and Wildlife based here in Wenatchee, Washington. So I did not have to travel probably nearly as far as a lot of you. I'd like to thank my co-author on this, Chris Repar, and then also like to acknowledge and thank Katie Shelby, my supervisor, and Michael Hughes for being able to bounce some questions off them as I run through this. So to start things off, I'll run through a quick introduction to the Mid-Columbia Recovery Unit, and then we'll jump into some of the specific movement patterns that we see of bull trout in the Wenatchee Basin. And then we'll sort of zoom in and look at some specific life history characteristics of bull trout specifically in the Chihuahua Basin, and then sort of wrap things up and go from there. So the Mid-Columbia Recovery Unit, sort of shown there in gray, is a pretty big swath of land. It covers basically all of central Washington, creeps on over into Idaho a little bit, and then comprises most of northeastern Oregon. It's important to note that bull trout were listed for protection under the ESA in 1998, and there are sort of 24 core recovery units or core areas within Mid-Columbia. So the Wenatchee Basin as a whole, obviously you're all sort of right at the confluence of where it touches into the Columbia, but for my study I sort of broke it up into 3 main reaches based off of some geomorphology. The upper reach is basically Tumwater Campground and up, if you know that area at all. The middle reach is sort of Tumwater Canyon itself, which is more higher gradient areas. And then the lower reach is sort of everything below that. So I'll be using a lot of pit tag data for my movement distribution stuff. Some of the main things to remember with pit tags, though, is that the detection probability can change based off of the array location. So based off of seasonal impacts, either the water stage or just the specific site itself, is you might come into slightly different detection probabilities. It also sort of is more of a course movement. The fish actually has to be detected at a site to know it passed by that area. For example, we have fish that might use the lower, or might use the Columbia River, but if they're only detected at the lower Wenatchee array, we can't definitively say that. And then, like I was saying, the data sort of only comes from fish that move. So a lot of the fish, especially with bull trout, who can express sort of a resident life history type, we may not glean a lot of information from them. However, pit tags do have a heck of a lot of positives. One of the main things that you've seen in a few of the other talks is that there is a lot of infrastructure in the upper Columbia. Basically, every tributary has a pit tag array. Most of the major tributaries have multiple pit tag arrays. So the Wenatchee, the Iniat, the Methow, they'll have a lower, a middle, and an upper array. And then all the main stem Columbia dams also have a pit tag array as well. So there are a lot of detections in places that we can find these fish. And then what it sort of lacks in resolution, it makes up for in sample size. Pit tags are extremely cheap and easy to deploy within a fish. Telemetry studies are a lot more expensive. Each tag can be hundreds of dollars. Pit tags, you can buy them for a couple bucks. They're very easy to install on a fish. Oftentimes we already have these fish in hand, so it's sort of an opportunistic ability that we have to sort of collect a lot of data. And then it is also really cool because there is no sort of stereotypical battery life. Like with a lot of telemetry tags that work on sort of days to months, pit tags will last for the entire life of the fish. So we can tag a 70-millimeter fish coming out of the Chihuahua and be able to track it over the next decade as it moves around the basins. So we get really cool information that otherwise we may not be able to get from a telemetry study where its scale, its sort of size, or its sort of time span is measured in months. We can measure movement in years. So I'll be showing you pit tag data basically from close to the last 20 years. We've tagged just over 5,000 fish in the Wenatchee Basin. Something to sort of keep in mind though, the vast majority of these tags have all been put into fish coming from the upper basin. And more specifically, the vast majority of these fish have all come from the Chihuahua River. So the Chihuahua River has a really healthy population. We also are able to collect bull trout at not only a weir that we have on it, but also a smolt trap. So we get them from a host of different places, but most of our tags come from the upper basin, and most of them come from the Chihuahua. So to jump right on into it, lower basin fish, we've tagged 408 bull trout in the lower basin. However, we don't have a lot of detections off of these fish. If you sort of look back at the data, however, most of those fish that were tagged were in the upper icicle or upper Pashastin that were probably more of the resident form, so you wouldn't have expected to get many detections off those. But of the fish that did move, we got 2% moved up into the upper river, and those fish both went into the Chihuahua. 17% of the fish moved into the middle reach, with the vast majority of those fish going into Chewaucum Creek. And then we had 15% of the fish move down and get detected in the Columbia. And like I said, though, it's sort of difficult with pit tags, and if you also include detections at the lower Wenatchee array, which is pretty close to the confluence of the Columbia, so it sort of makes sense to assume that those fish also went out and used the Columbia, just weren't detected at a dam. The number actually increases to about 26% of the fish tagged in the lower basin were assumed to possibly go out to the Columbia. And we see a lot of that movement pattern that is consistent with sort of overwinter movement, where fish are either detected dropping down below like Rock Island, or during the sort of late summer, late winter, early spring, getting redetected on the adult fish ladders coming back up. And then we also, bull trout are a cold water obligate species. So the Wenatchee gets fairly warm during the summer months, well in excess of 20 degrees C. So during the summertime, we see a lot of movement of fish, bull trout moving to sort of colder tributaries, seeking out cold water, sort of thermal refuge. As we move into the middle basin, again, we have a decent number of tags out, 821. Most of those have been put into fish at Tumwater Dam, but we have a heck of a lot more of these fish with detection histories. 88% of these fish have detection history, so much larger sample size. And we see that about a third of these fish move into the upper basin, with 20% going into Chihuahua, 20% going into Nason, 20% going into Chihuahua, 11% going into Nason, and only 1% getting detected in the white. And then we have the vast majority of these fish tagged at Tumwater move into Chewaucum, we see both the sort of movement patterns that would be consistent with spawning, as well as fish seeking out thermal refuge, so getting detected not only during the heat of the summer, but also sort of during the fall when they'd be spawning. And then we see 18% of the fish moving down to the lower basin. Most of those fish go into the icicle. Again, you see sort of seeking out a thermal refuge, as well as some overwinter movement there. And then 3% of the fish tagged in the middle basin are detected out in the Columbia. When you include detections from the lower Wenatchee array, that number increases to about 11%. So a few fish going down to the Columbia. And again, we see a lot of movement, sort of fish seeking out thermal refuge. As we move into the upper basin, we have a lot larger sample size. About 3,800 tags been put out over the last close to 2 decades, and we have detection histories from about 52% of those fish. And what we see is upper basin fish stay in the upper basin. 95% of these fish are not detected anywhere else. They are only detected in the upper basin. 3% of the fish generally will move down to the middle with either detections at Tumwater or up in Chewacum. And then only 1% are detected down in the Columbia or assumed to be down there if they're also detected on the lower Wenatchee array. Also of note, and what I thought was kind of surprising, is these fish really aren't detected in multiple tributaries. Only 17% of these fish are detected in 2 or more tributaries. They seem to use whatever their tributary that they are either natally spawned in or where at least they're rearing in, and then they sort of exhibit a lot of sort of rearing in the main stem Wenatchee, but they really aren't bouncing around from the Chihuahua to Nason to Chewacum. They really just use one tributary. And then we see a lot of movement that's sort of consistent with overwintering in the upper Wenatchee or the Lake Wenatchee itself. So some kind of cool stuff coming out of that. So again, to sort of rehash it, upper river fish generally stay in the upper river. Middle fish generally stay in the middle river with some movement out to the Columbia or lower river. And then the lower river fish generally stay in the lower river, but with about a quarter of the fish moving out into the Columbia. And again, this is not adjusting for detection probability at the arrays, so this is just straight detections, which is important to remember. And next time you're trying to impress somebody with some cool bull trout statistics about the Wenatchee, our oldest fish is assumed to be over 16 years old. It was tagged as an adult at 550 millimeters, and then we had detections off of it for the subsequent 12 years. So that is a very old fish. Our largest that we've collected is 850 millimeters. That rivals some of the Chinook that we have returning. And then some of these fish do move a really long ways. We had a fish tagged at Priest Rapids last year, get recaptured at Tumwater Dam. And then we had a fish a couple of years ago, a juvenile tagged at Chihuahua, that was detected up in the Okanagan. So these fish can move large distances. And so there's sort of 2 parts to this study, or at least to my little story here. Another part of it is sort of focusing in on the Chihuahua. There's a lot of research that's coming out on adult bull trout, but very little on juveniles. And one of the cool things with our Chihuahua trap is we're able to collect some of those juveniles. So I'm going to share some of that information. We've run scales off about 1,500 fish, and we see that of the fish immigrating out of the Chihuahua, 6% are age 1s, 70-ish% are age 2s, 24% are age 3s with a few age 4s in there, but the vast majority of them are age 2s. And then because we can track these fish for their entire life, once they are tagged going out as juveniles, we get 21% redetected back in the Chihuahua the next year. 10% have their first redetection after 2 years, and then that jumps to 35% getting their first redetection after 3, and 27% getting in their first redetection after 4. And so when you sort of look at that, the 2-year-olds that are going out generally will spend 4 years out in the Wenatchee, and the 3-year-olds generally spend 3 with everything returning sort of assumed to be mature at roughly 6 years old. And then once those fish are assumed to be reaching the sort of age of maturation, 6 years, we see that 73% will be detected back in the Chihuahua annually thereafter. And then 17% get detected sort of every other year, so possibly like a skip year spawning or for whatever reason only showing back up in the Chihuahua every other year, with the remaining 10% showing some combination thereof. So the take-homes here are most of the fish immigrate out of the Chihuahua at age 2 with some 3s. They rear for 3 or 4 years in the main stem Wenatchee with basically no detections anywhere. They're not moving around, not moving over arrays, and then they get picked back up at age 6. Once they are mature, they are detected annually from thereafter, so it's a pretty strong pattern. And then, one of the sort of themes of this meeting is scale matters, and it really, I think goes to show if you are trying to restore or work with the species, it really matters depending upon what your area of interest is. We see movement patterns differ where you are in the basin, Chihuahua versus Wenatchee versus the upper Columbia as a whole. We also see that, fish in the upper basin generally tend to exhibit this sort of adfluvial life history. Fish in the lower river show a lot more fluvial life history. And that there are a ton of sort of still data gaps with juveniles, but a lot of, some of that information can be answered with the use of our sort of smolt traps and opportunistic tagging. And so sort of what's next from here, all of this is just opportunistic. My main sort of area of interest is Chinook, and so we're out there with our smolt traps and our electrofisher. We're out collecting Chinook, but at times we will get bull trout. And so continuing to be opportunistic in how we're able to sort of work with this data, analyze it, is really important. And then, I think it'd be cool to look at sort of estimating tributary assignment or sort of natal rearing. They seem to be showing a strong sort of fidelity to where they were probably born. And so seeing if you could use pit tag detections to sort of look at natal rearing tributary would be a pretty interesting thing as well. And then one of the really big important things is, collaborate with you all. A lot of this stuff is all done opportunistically. And I think, this conference is a great way to sort of bring everybody together. And far too often we end up siloed in our own projects, our own data, and we forget that there are other entities doing basically the same thing. And we're all out here to try and conserve and restore the stock or the species, and we can really benefit from working with one another. So thank you all for listening. And with that, take any questions at the end, I think.
All right, our next talk is by Jose Vazquez of U.S. Fish and Wildlife Service, and his talk is titled Assessment of Potential Bull Trout Extirpation in the Nason Creek Watershed. So is Jose here? I thought I saw him earlier, maybe I didn't. Oh, okay. Just take a few deep breaths, maybe. Get some meditation time or something.
All right, hello everybody. Okay, so for those of you who don't know me, my name's Jose Vazquez. I am the bull trout research biologist for U.S. Fish in Leavenworth. And today I'm going to talk about one of the projects we've been working on for a few years now, which was an assessment of potential bull trout extirpation in the Nason Creek watershed. All right, some of this is going to be a rehash of what Josh was just talking about, but I'm going to start off by kind of giving an introduction to Wenatchee Basin bull trout populations, just for those of you who aren't familiar with them. So we have 8 recognized populations within the Wenatchee Basin. You can see them on the map here. I'll go over them in a sec. So 2 of them are doing particularly well. Josh was just talking a lot about the Chihuahua population. That's a very stable population. By bull trout terms, we typically see over 400 reds annually. That's a lot for bull trout. White River is also pretty stable, usually has around 100 reds per year. But most of the other populations are kind of a different story. So 4 of them, we're not necessarily sure what the status are. Icicle, the 2 Pashastan populations, and to a little lesser extent the Chewacum. We think these populations have a combination of those migratory and resident life histories. And the resident life history are hopefully keeping the populations fairly stable. But it's hard to measure the size of a resident population, so we're not necessarily sure, particularly for some of the Pashastan populations. Like we think that Etienne might be doing poorly, but again, we haven't been in there for a while and we don't really know. We do know, though, that 2 of the other populations, the Little Wenatchee and the Nason Creek population, are not doing very well. We have a lot of data on the Little Wenatchee and Nason. I'll circle back to Little Wenatchee at the end of the presentation for a bit, but we're going to focus most of our talk today on Nason Creek. Okay, so everyone here has seen Nason Creek at some point, probably. It's the creek that flows between Lake Wenatchee and the top of Stevens Pass. It follows the highway for most of its stretch. It has a lot of bull trout habitat, but a lot of it is impaired. The highway and corresponding development has a lot of impacts. Hopefully on the picture there, yeah, you can see that there's a culvert that we took a picture of in Mill Creek that covers about 50 meters of what would otherwise be prime rearing habitat for bull trout. So there's a lot of impacts throughout the available habitat. Also, a lot of the habitat within Nason Creek is fairly climate sensitive. As we're switching to a rain-dominated system, we're seeing that a lot of that habitat is warming, a lot of it's having lower flows in the summer, both of which are not good for bull trout. Habitat's also fairly limited, or I should say that the available habitat is fairly limited. There's a lot of barriers within a lot of the tributaries that are colder that you would otherwise expect to see bull trout, cutthroat, other cold water species, but because of the barrier falls, they aren't able to access them. Now that said, there still is a unique population within Nason Creek. The population has primarily spawned within Mill Creek and White Pine Creek that you can see circled on the map there. That's from about 20 years worth of red surveys that we know that those are the primary spawning areas. They have spawned within the main channel of Nason Creek as well, but the red surveys indicate that most of the spawning has been within Mill and White Pine. Now that said, WFW has been doing surveys for the past few years, and data is indicating that those populations have really crashed. So White Pine, you can see from that figure there that we haven't seen any reds within that system since 2019. Mill Creek, we haven't seen any bull trout reds there in over a decade now. So again, those are the primary spawning and rearing areas, at least we think, for Nason Creek. So that raises the question, is the Nason Creek bull trout population still extant? And also the question, if it is the population spawning elsewhere, and what is its current status? So we wanted to answer these questions, so we came up with a study that had the following objectives. We wanted to determine if the Nason Creek bull trout population is extirpated, and also assess the distribution and status of the population if it is still present. So in order to do this, we adopted a three-tiered strategy. We first started out by completing census snorkels on all of the known bull trout spawning habitat within the historic areas, Mill and White Pine. Then, based on those results, we then expanded to an all available habitat within Nason Creek that bull trout can access. We collected eDNA samples from all of those to basically see, okay, if they're not within Mill and White Pine, are they elsewhere? And then we followed up those eDNA surveys with snorkel surveys within high eDNA concentration areas in order to assess the densities at those locations and also confirm that they were in fact present. So I'll go into that more as we keep going, but for right now, we're going to focus on that first part of the three tiers where we were going in and snorkeling Nason Creek and White Pine to assess the populations there. So we did night snorkel surveys during our census, and we use these because it's a really powerful way of assessing all bull trout age classes. So night snorkel surveys have high detection probabilities of young of the year, which a lot of our other fishery surveys do not, as well as juvenile subadults and to a certain extent adults. So we use those relatively powerful surveys to survey, again, all potential habitat within Mill Creek and what we could access within White Pine Creek. So that ended up being about 1 kilometer in Mill Creek. We sampled all available habitat up to a barrier falls that they don't get above. And in White Pine Creek, we surveyed the first 800 meters up until a high gradient section that ends in a barrier falls. We didn't get that high gradient section, but we got the primary rearing habitat within that creek. And then during the snorkel surveys, we also captured bull trout. We did a genetic assessment on those. Unfortunately, I'm not going to get into much of those results today, just in the interest of time, but I'll touch on a couple key ones as we keep going. Okay, so here you can see again, we surveyed all of Mill Creek and most of White Pine Creek circled there. I'm going to bring up the results right now. So the bottom line from the snorkel surveys is that we did find bull trout within these creeks, but we didn't find very many. We found a total of 15 fish in all of the spawning and rearing habitat, or most of it, between Mill and White Pine. We only found one juvenile in each of the creeks, and most of the rest of the fish were older subadults. So there's a couple conclusions that you can draw from this. The good one is that the Nason Creek population is still extant. Again, we did some genetic analysis. All of the fish that we captured came out as Nason Creek fish, so the population is still out there, which is good. But that said, you know, these were very low densities, particularly of juveniles, and it indicates that very little spawning and rearing is happening in those historic spawning areas. So that then raises the question, okay, so if they're not spawning within the historic areas, are they anywhere else? And that's what we used eDNA for. So this is a picture that shows all of the eDNA samples that we collected during the study. We limited sample collection to the available habitat within Nason Creek because we've already collected elsewhere. We know that that is where the bull trout typically are. And we use the Rocky Mountain Research methodology, which is collecting eDNA at 1 kilometer intervals, collecting 5-liter samples. And we use this because our office and other labs have shown that this is a really powerful way of detecting juvenile rearing habitat. Basically, if you have a sustainable population of juvenile bull trout and you're using eDNA best practice methodology, you should detect them using this protocol. So, in addition to this, we quantified the eDNA within all of our samples, which basically let us get relative densities of eDNA that we could compare to see which sites had the most eDNA. There's a lot of caveats when you do eDNA concentrations, and I'll get into them in the results on this next slide here. Okay, so the dots all correspond to our eDNA sites. The green circles indicate that we found eDNA at a location, and the circle size corresponds to how much eDNA we found within those sites. Now, again, you have to be careful when you're looking at eDNA concentrations. There's a lot of things that can affect them. So, for example, everything from fish metabolism, how close you were to the fish, to a lot of environmental factors, temperature, solar exposure, microbial activity, bed interaction, the list goes on and on. So just because you saw, let's say, 100 copies of DNA at one site and 1,000 copies at another, that doesn't mean there's 10 times the amount of fish necessarily. But if you're sampling a uniform system like Nason Creek and you collect a large amount of samples like we did, you can draw some general trend conclusions. So you can see up there in the upper portion of the watershed, a relatively cold area, we found a lot of eDNA at several nearby sites. That indicates that there's something that we should check out going on there, which is what we did with the third set of methods. So we then went into these sites with the night snorkel surveys and basically surveyed at those spots where we found a lot of eDNA. So we did 200-meter night snorkel surveys at each of those reaches. And then here's a map of the reaches here. You can see, again, we surveyed the reaches that were up top in the colder habitat. If you saw on the last map, we did find some bull trout eDNA further downstream, but those are in relatively warm areas, and we think that that's metapopulation holding habitat, which is kind of a different discussion. I'm just going to focus on the rearing habitat eDNA sites for right now. Okay, so the results from those snorkel surveys, if I can go to the next slide. Anyone have a different clicker? Yeah, okay, I'm gonna have to tell you to click a lot then, sorry. Yeah, that's fine. So, okay, we didn't find any bull trout DNA within the first 2 sites that we snorkeled, the 2 red dots that you can see there. But luckily, on the last 2 nights, we did find a total of 20 juveniles, 10 at each of the sites. Can you click again for me? And just go ahead and click through the whole box. It'll fill up. All right. So I'm spoiling a surprise, but basically the conclusions that we found from this is that bull trout were spawning and rearing in Nason Creek, right? So we found 20 juveniles. That is indicative of successful spawning and rearing. This was an area that we hadn't necessarily found bull trout in before. At least we hadn't found any reds in before, but it does look like they're spawning and rearing there. But that said, that's a good result, but these were really low densities for the methods that we used. We used the scientific equivalent of a bloodhound, basically, to sample the entire watershed, look for the spots where we could find the most eDNA. We went to those spots, we used the most powerful methods that we have to look, and we found a total of 20 fish. And what results, we haven't processed these genetic results yet, but what results from other watersheds, including Nason, or parts of Nason, are showing us is that these fish were probably related. So it doesn't look like there was a lot of spawning going on there. I can't give you a number as to how many adults were spawning, but it doesn't look like there's a lot of activity. So the population is still extant, but it looks very small. How sustainable that is a good question. And if you can go ahead and go to the next slide. Can you click twice? Yeah, we'll start with those. One more. There we go. Okay, so right now I want to put all of these conclusions together into one slide because we had a bunch of different parts to this. So the first conclusion, arguably the best one, is that the Nason Creek population is still extant, right? And then we also found that spawning seems to have transitioned to Nason Creek, right? The majority of spawning appears to be there. We're not finding any in Mill and White Pine at the moment. And then click through the depressing stuff. One more. There we go. Okay, so again, our densities, though, were really low, right? And it's questionable whether or not these densities are sustainable in the long term. And I'll also say this too. I know I saw a few people from the Yakama in the room right now. We have some density data from there and other populations that have extirpated that use less powerful techniques than we employed here, and they recorded higher densities before those populations became extinct. So this is not boding well for the Nason Creek population. I can't say definitively that it's definitely going to become extirpated soon. I can't give you a definitive time frame, but our research is really pointing to the days may be numbered on this. So that raises the immediate question, can anything be done to preserve the population? And I will take 3 clicks. Thank you. Okay, so maybe is the answer. Obviously, habitat restoration, this room knows what that can do. Also, introduction to upstream areas is something that we've had some success with in other bull trout populations. Mill and White Pine Creek, we think, have good habitat upstream of those barriers, so by moving fish up there through translocations or barrier alteration, that might increase carrying capacity that could help the population. Captive rearing in the Yakama Basin, there's been a lot of success with capturing young of the year, raising them up for a year, and then releasing them. Your survival can go up literally 100-fold in some cases. And I will take 3 clicks now. There we go. Feasibility is the big question on this, right? If I was going to have everyone here raise their hand and ask, who primarily studies salmon, who primarily studies any other species, we know which one's going to win, right? There's not that much bull trout funding out there, and whether or not a lot of these things are feasible is a good question. And then you also have to ask the question, is it already too late, right? We didn't find very many fish, and like I said, genetic diversity is a concern. I don't know if we would be able to raise a sustainable population off of what's left in Nason Creek. We might be able to. Again, I can't say either way for sure, and we're still waiting for some of those genetic results, but what we saw wasn't very hopeful. So now I want to go to kind of a big picture scale slide and bring it back to kind of the populations we're talking about at the beginning. Nason Creek is not the only near extirpation Upper Columbia bull trout population. 1 click this time. Thank you. Okay, so in the Wenatchee Basin down there at the bottom, we know that at least 2 of the 8 populations are at risk, right? We just went over Nason Creek. Little Wenatchee that I mentioned earlier might be even worse. And again, we don't really know what's going on with Etienne or some of those Peshastin, or the other Peshastin population. Entiat Basin, the Mad River, and the Entiat both have a lot of issues. The Entiat in particular is up against a barrier. It's in warming habitat, and there's brook trout there. That's not very good for the long term. Chelan's already extinct, and then in the Met Howe, most of those populations are at risk. Bull trout red surveys that John Crandall and others have been doing recently found less than 11 reds in 6 of the 7 populations that they were surveying, at least within surveyed reaches, and then a couple of those reaches have zeroed out. We started another preliminary extirpation assessment on one of those populations, Chewuk, and this year in the rearing habitat, we found lethal temperatures and 3 fish total. So things are not looking great. Nason Creek is not alone. 1 last click. Okay, so the message I want to leave you with is that maintaining current Upper Columbia diversity levels, if we choose to do so, will likely require near-term recovery actions, right? We might already be, 5 years too late for some of these populations, but if we decide that we want to preserve the current level of diversity, we're probably going to need to address some of these issues in the immediate future. All right?
All right, our next speaker is Jeff Caisman with the Yakama Nation, and his talk is titled Trials and Tribulations, 25 plus years of coho salmon reintroduction.
I was actually told I should have named this Trials and Tributaries, so pretend that I was smart enough to come up with that title, and, oh, awesome, thanks. Does this work? Cool. So what I'm going to talk to you guys about today is kind of a general overview of Yakama Nation's reintroduction efforts for coho in both the Wenatchee and Met Howe basins that's been going on for probably 25 plus years. So we've got the Columbia River Basin here. You don't need me to talk about that, but you obviously know it used to support large runs of salmon, historically averaging 7 to 16 million of adult salmon returning annually. We also know that this was an important source for food for tribes with an estimate of 22 million pounds of salmon harvested annually by tribes. So zooming in a little bit to the mid-Columbia, where we're going to be working in the Methow and the Wenatchee, we knew there were large runs of coho returning to these two basins, with 23 to 31,000 annually returning to the Methow and 6 to 7,000 returning annually to the Wenatchee. These populations were decimated in the early 1900s, extirpated by the 1990s due to the standard cast of characters, water diversion, habitat loss, overfishing, you name it. So the goal of Yakama Nation here when we set out to do this was to reestablish naturally spawning populations of coho in the Wenatchee and Methow basins, as well as open opportunities of these returning coho for tribal harvest. So we took a tiered approach when facing this challenge. Our first step was this broodstock development phase where we have no mid-Columbia broodstock, so we need to draw from these lower Columbia donor populations and kind of adapt that to a locally adapted population for our mid-Columbia. These steps have been completed in both the Wenatchee and Methow basins. Next was broodstock development phase 2, and this is where we wanted to encourage local adaptations within our basins by encouraging adults to move upstream and inhabit some of these reaches where we haven't seen them yet. This is accomplished in the Methow. We're still working on it in the Wenatchee. And then the final step was this natural production phase where we just want to continue to encourage this upstream dispersal of returning adults, as well as starting to rely more on natural production and kind of phasing away from hatchery production. So the Yakama Nation launched a feasibility study in 1996 to determine if we could take these lower river donor stocks and have them locally adapted for longer upstream travel in the mid-Columbia, and that was determined to be feasible. And so again, our objective was to develop this locally adapted, naturally spawning coho stock in the Wenatchee and Methow basins with a 3-year mean escapement goal of greater than 1,500 wild fish, as well as a 23% harvest rate that includes mixed stock, terminal, and main stem harvest. These are our basins we're working with here. We have the Methow Basin and the Wenatchee Basin. Again, Methow Basin is a step ahead, working on the natural implementation phase, while the Wenatchee Basin is a little one step behind, and we'll talk about why that is here in a minute. These are the performance indicators we're using for some of our monitoring and evaluation. I'm not going to talk about all of them, but just kind of give you an idea of what we're looking at. We'll talk about smolt to adult returns. We'll talk about escapement and distribution, and a couple of these other things. So overview of our production efforts. In the Wenatchee Basin, we have 5 in-basin acclimation ponds where we rear fish, as well as at Leavenworth National Fish Hatchery. We spawn about 500 females annually, which equals about 1.4 million eggs and results in about 1 million smolts released annually in the Wenatchee Basin. In our Methow Basin, we have 7 in-basin acclimation ponds, as well as rearing fish in the Winthrop National Fish Hatchery. We spawn about 325 females every year, which equates to about 900,000 eggs and 700,000 smolts released annually. So, quick overview of some of our monitoring methods. I'll just glaze over these real quick. We have some smolt traps where we're enumerating out-migrating juveniles. We do spawning ground surveys where we're looking at red distribution, as well as taking carcass samples. We've done snorkeling surveys in the past, looking at juvenile abundance and distribution. And starting in 2019, we started pit tagging returning adults at Priest Rapids Dam to give us a better estimate of escapement as well as in-basin distribution. So what the rest of this talk is going to be about is I'm going to talk about some of the challenges we've faced in this reintroduction process, as well as some of the ways we've overcome these challenges or how we're still working to overcome them. So challenge number 1, and I mentioned a little bit earlier, is adapting these lower Columbia River stocks that aren't used to longer upstream travel and adapting them for further upstream travel in our mid-Columbia populations. Then we want to expand the in-basin distribution of coho, make sure that they're using all or as much as we can available spawning habitat. And finally, one of the main goals of this was to open a fishery, so increasing escapement numbers to the point where we feel comfortable opening up a fishery for harvest. So challenge number one, right? We have no more broodstock from these historical mid-Columbia populations, so we have to use this lower river Columbia donor stock. And the difference here is some of these lower stocks, they might travel 150 miles to their spawning habitat, while the Wenatchee River is at Columbia River mile 468 approximately, and the Methows at 523. So we have these fish that are used to this shorter travel time, and we want to see if we can develop this stock to make these longer upstream travel that requires significantly more energetic costs. And so we set up an experiment where we had 4th and 5th generation Wenatchee River stock that was spawned in Basin, and we wanted to compare it to this lower Columbia River donor stock to see if we had seen local adaptation to this longer upstream journey over the course of 4 or 5 generations. And what we did is we did 3 years of side-by-side releases with these groups, totaling about 500,000 smolts, and we compared smolt to adult ratios between our lower river donor stock and our locally adapted Wenatchee River stock to see if we were seeing local adaptation. And so these are the results here. I'll orient you to these graphs real quick when I pull them up. On our y-axis, we have smolt to adult ratios. On our x-axis, we have our release years. In blue is this lower river Columbia donor stock, and in orange, we have our Wenatchee stock. And so it really just pops right off the graphs here that you can see we had significantly higher smolt to adult ratios for our 4th and 5th generation Wenatchee stock. So it shows us that we are seeing this local adaptation, that over the course of 4 or 5 generations, this lower donor stock has adapted somewhat to this longer upstream travel. And this also shows us that, okay, we've shown that we can make this local adaptations between basins. Now we want to see if we can do that within basins, right? Encourage adults to migrate further upstream and inhabit new reaches to use all available spawning habitat. So, right, how do we expand in-basin distribution of coho? One of the ways we do this is we move our collection sites further upstream to basically select for those individuals that have made that further upstream journey within the basin and are inhabiting some of those unused spawning reaches. And then also moving acclimation ponds further upstream to encourage returning adults to, like I said, get at some of those unused habitat reaches. And so I'll go over this one real quick. This is an example in our Methow. Hope you can see it, but in red we have red distribution, and in black we have our acclimation sites. And so if you go through it real quick here, you see the expansion of this red distribution and increased red distribution further upstream as we push some of those acclimation sites further upstream. So again, this shows us that we've accomplished increased dispersion of returning coho by moving some of these acclimation sites further upstream. We're working on this same thing for the Wenatchee, but we have a little bit of an issue in Wenatchee. So on the left there, if any of you rafters are familiar with it, that's what the rafters call exit drop, and that's just downstream of Tumwater Dam and Tumwater Canyon. And this is a summer flow picture, and it doesn't look too crazy. But in the fall, when coho are coming back to spawn and the river comes up, that develops a velocity barrier for returning adults. And when that velocity barrier does set up, it basically blocks adults from inhabiting all of that upstream spawning habitat. So you can see in that red circle, that's a pretty big chunk of spawning habitat that's potentially being missed because of this velocity barrier. And this is a springtime GIF here of that exit drop, and it doesn't quite get that crazy in the fall, but it just kind of gives you an idea of the potential for that little section as a velocity barrier, and obviously how it would be difficult for fish to migrate up that during the fall. And so this is, one of the things we wanted to do is expand upstream collection and collect fish that already had ascended that velocity barrier, therefore making it more likely they'd pass on that trait to progeny. So on the y-axis, we have percent of broodstock collected above that velocity barrier. And what you can see here is we've had an overall increase in number of fish we've collected above that velocity barrier for use in broodstock, but it's also variable due to seasonal flow conditions. Sometimes we get high flows early in the season and we don't get many fish at all that are moving up there. So collection is a little bit finicky depending on what kind of flows we get that time of year. And finally, the last thing we wanted to do was increase escapement to the point where we could open a fishery. And so we have escapement, adult escapement on our y-axis. Our blue bars are the Wenatchee Basin escapement, and the orange bars are the Methow Basin escapement. And so what you can see is we've had a kind of trending upward in terms of escapement, especially in the last few years, where we have seen these large returns. In 2021, we had record returns for both basins with 27,000 and 25,000 returning to the Wenatchee and Methow respectively. And again, one of the main goals of this whole program was to be able to open a fishery so people could benefit from this resource. And we've had an on and off fishery since 2009. And the last 5 years on the Icicle River in the Wenatchee Basin, we've actually had enough of a return to open it for the public fishery as well. So it's been good for both our tribal fishery as well as our public fishery. So kind of wrapping things up here, some conclusions and where we're going next here. We've shown that local adaptation from these lower donor stocks is possible in the Wenatchee and the Methow. We want to continue brood stock selection for adults captured above tumwater to kind of increase dispersion and use of some of that upstream spawning habitat. We want to continue to move these acclimation sites upstream also to increase further upstream movement of our adults, as we've seen has been successful in the Methow Basin. And finally, we want to do some outplanting starting in 2024 just to kind of get some of those adults spawning in some of those unreachable upstream habitats. And that's all I got.
All right, moving along. Our next speaker is Tom Kahler with Douglas County PUD. I know Tom's here. I saw him. And his talk is titled Factors Facilitating the Recovery of Okanagan Sockeye.
Wow, those are bright. Okay, so I tacked on to my title to include factors necessary for population persistence. For those of you not familiar with the Okanagan sockeye population, this will be hard without pointing. Anyway, the yellow line there, of course, is the US-Canada border, and you can see on that border, north of that border, there's a great big lake, and it says Kelowna. That is Okanagan Lake, and that is the source of the Okanagan River. And there's a chain of lakes there. Okanagan is the top, Skaha is next, Vaso, and then the Soyuz Lake on the border. The basin is pretty much a mess with most of the stream or the river is channelized after the 1948 flood. They fixed it by putting it all in a channel. It has these 3 dams at the outlets of the lakes. There are, it's very much developed basin. It has a population of, I think, around 350,000, some major population centers, lots of agriculture, lots of uses for water. It's kind of like the Napa Valley of Canada, lots of wine production. In 1995, Douglas PUD went to Canada looking for mitigation opportunities. We wanted to get away from hatchery solution for sockeye mitigation, and we wanted to increase natural production of sockeye in the Okanagan. And we went up there, and the fisheries parties were not communicating with one another very well, and it took a while, but we got them to form what's called the Canadian Okanagan Basin Technical Working Group, cog twig. It was ob twig at that time. They added the C later because they also have a bob twig, which is a bilateral group. And in 1999, they formed that. And in 2000, early 2000, they gave us this short list of opportunities for mitigation. Their recommendation was to improve water management. And the reason for that was because they had a bunch of rules for operating the water system, releases from those outlet dams, primarily from Okanagan Lake. And you can see in that lower left for incubation, if you're outside of that green rectangle, you are either, if you're above it, you're scouring reds. If you're below it, you're desiccating them. And the challenges for the water managers, it wasn't that they didn't have a will to protect fish, they just had challenging conditions. They had this huge variation in annual inflow. They had multiple competing interests, lake levels, river flooding, agriculture, and so on. And that included recreation, which is insanely popular to float the Penticton Channel in various floating things. And then there's the third issue that they were dealing with at that time was that they didn't get along well. They didn't trust one another. The fisheries people didn't trust the water managers and vice versa. And so getting them to work together was quite a coup. And that was my predecessor, Rick Klinge, that accomplished that. But since then, we've had this basically a 30-year almost collaboration. And these are the partners. And they developed what was called the Fish Water Management Tool. This whole presentation is not about the Fish Water Management Tool, but I'm starting with that. And it's basically a model, a multi-model model that allows the fisheries and water managers to plug in water use scenarios, water release scenarios, and look at outcomes for fish and the various competing interests. And there's a bunch of real-time and near real-time data, and it's a collaborative decision-making process instead of just the water managers. Now the fisheries folks are involved, and they work well together. It's been a great collaboration. And here are the rules, really, between sockeye spawning and fry emergence. The rules are keep flows between those two green dashed lines. If you get above them, you're going to scour reds, if you get or alevins, and if you get below them, you're going to desiccate them. And this is a real life scenario, and you can see there's 3 spawning cohorts all heading towards, moving to the right, heading for that black line, which indicates emergence. And if you can get them to that point without getting the water out of those, above those, that row of yellow triangles that are faint, you can hardly see, then you get a gold star and you've achieved your goals. It's worked very well in providing increasing fry production per female, and it's worked very well for the incubation phase. For the fry emergence phase, it's a little dicier because you have a freshet that's starting to build, and the guys need to decide whether you let the water rip or not. And so they're monitoring fry emergence. And there were a couple years there, 2019 and 2017, where the flows were just too big that didn't give them the decision space that they needed, and they had to let the water go and did some scouring. And what the result has been is numbers of pre-smolts per female has increased from a pre-fish water management tool era of 37 to up to 69 in the post era. There's been a dampening of the variance in pre-smolts per spawner, and that's resulted in, actually now there's a statistical relationship between spawners and pre-smolts, whereas those gray boxes are the pre-era where there was no relationship. That's just a median log line. I don't know what that's doing. I think I did something wrong here. There we go. Good, that's a relief. The result has been an increase in adult returns, and the mean for the 30 years before that was 30,000, and the mean now is greater than 200,000. But there's been other factors. It's not all been the fish water management tool. During this same period, there's been a reintroduction program going on. There's been restoration of passage to habitat that's been blocked for a long time. There's been habitat restoration. So I want to talk about that. And this is a few slides from a presentation that David Duvall from Grant PD gave at the AFS Western Division meeting in Boise last spring. And when we arrived up there in 1995 to talk with the Okanagan Nation Alliance, they had this vision for a reintroduction and restoration program, and they were happy to hear that we wanted to be a partner in that. And other partners arrived shortly thereafter. Grant and Chelan arrived, and they have been working on this Put the Fish Back initiative with the Okanagan Nation Alliance, which is reintroduction of sockeye into Skaha Lake and ultimately to Okanagan Lake. And it is a 12-year, it was a 12-year experimental reintroduction. It was highly successful. Chelan and Grant built and fund the operation of this hatchery, which I can't pronounce, so we just call it the Penticton Hatchery. And they produce fry for outplanting to the lakes. And they started the first outplanting in 2003 with fry produced from the Shuswap hatchery. And since then, there's been other developments. McIntyre Dam, the PRCC funded the restoration of passage there. Then the Colvilles and ONA restored fish passages to Skaha Lake in 2014, and more recently to Okanagan Lake, and including the political barrier to access as well. And so here's the fry production over the years, beginning in 2003, and it built slowly and concentrated initially on Skaha Lake, and more recently with larger releases focusing on Okanagan Lake. And the same, concurrent with that has been habitat restoration efforts. Here's a specific project that was intended to increase spawner habitat for the Osoyoos population. Likewise, for the Skaha population, there's really not spawning habitat available other than in the Penticton Channel. And so they built spawning beds to accommodate those spawners, and those have been very successful. So here's the escapement that's resulted from all these activities, and you can see over time with the blue, we've started to get Skaha Lake production that's contributing pretty substantially to adult returns, and also more recently the Okanagan as well. And this has, it doesn't necessarily match the dam counts, the well's dam counts, so we see escapement being a fraction of the dam counts. And there's a reason for that. This is historic data from 1967 through 98, and the blue is the wells count, and the orange is the resultant escapement. And what you see is a pretty big gap in those. And Kim Hyatt and, the black line is the proportion of the wells count that the spawning escapement represents. You can see it's all over the map from as high as nearly 1 to greater, just over 4% in some cases. So that great variation is a result of thermal barriers that set up nearly annually in the Okanagan River. And Hyatt and Rankin had that in mind when they developed the pre-fishwater management tool Canadian escapement objectives, and those were in both wells dam count units and spawner count units to represent the difference that they expected. It's roughly half. So they were expecting quite a bit of interannual variability, but the median was approximately half the count at wells is going to show up on the spawning grounds. And this was first described by the Bureau of Commercial Fisheries back in the 1960s. They recently constructed Rocky Reach Dam, and they wanted to know if that was going to affect Okanagan escapement. And they didn't find an effect of Rocky Reach Dam, but they did discover this 70-degree magic number that fish stopped moving at. And so here's 2015 as a kind of like their worst case scenario, and you can see in this that the river temperature in the lower Okanagan got to over 22 degrees, which is our magic number in Celsius, early on, actually before the sockeye even arrived at wells. And it persisted for quite a while, and there's a little dip in there that a bunch of fish made a run for the border, and it's more than a day's trip, and so a lot of them didn't make it, and there was videos of the carnage that went viral. One of the problems is the water coming off of Osoyoos Lake is quite warm, and it's warming up earlier than the lower river, which is influenced by similkameen input. So fish will head up the river, get to the confluence of the similkameen and Okanagan there at Oroville, and they're like, I can't go into the Okanagan, I'll turn into the similkameen, it's cooler, but then that eventually warms up, and that is a death trap for them. So looking at Lake Osoyoos here, there's a south basin, central basin, and north basin. Those look like this. Only the north basin has a usable volume between the hot thermocline and the anoxic hypolimnion, and that gets squeezed throughout as the summer progresses. This is getting worse over time. Kim Hyatt was expecting that eventually Soyuz would become unavailable as a rearing habitat, and I see that's probably in the future here with climate change. But it's not just the thermal barrier in the Okanagan. It's now we've seen thermal issues in the lower Columbia as well. On the left, that's 2015. 17 degrees C is above that. It's problematic for sockeye migration. Not so much for the Okanagan stock because they're adapted to hot water, but it's still a problem. And when you get up around 20, it's a big deal for all the stocks. And 22, they just stop moving. And you can see in 2015 that it was already up into the danger zone in the early part of the run. And in 2014, on your right, is a more normal year. It didn't get to 17 until halfway through the run, and it never really got to 22 until after the run was past Bonneville. All right, so here I had shown you something similar. This is 98 now through, or 99, sorry, through 2022. And again, there's that ratio of escapement to dam counts. Prior to, well, from 99 through 2011, it was about 61%. Starting in 2012 through present, the average is 29%. So what's going on? It's a combination of things. One of the things that's happened since, with the resurgence of the Okanagan population, has been a resurgence of harvest. In the lower river, harvest increased beginning around 2009, and there's a statistical change point there. Also, a fishery developed above wells. There really hadn't been a fishery for sockeye, and now there's a robust fishery for sockeye, and it's a pretty substantial removal. So when you pile that onto this thermal barrier annual variation, you get the combination here. Now there's a post fish water management tool escapement objective that's 138,000. That's for the Okanagan, the Soyuz stock, and Skaha. It does not include Okanagan Lake, but it needs to. And look, we've only achieved it twice in history. And I've added total US harvest to this. There's Canadian harvest I did not add to this. Other effects of harvest in the lower river gillnet fisheries, there's actually size selectivity going on, and that's a problem for Okanagan stock because they tend to produce a lot of one ocean fish, and they have low fecundity. So, for example, you need 6,600 brood to seed the hatchery with a 23 to 2,400 eggs per female. So the fish water management tool results to date, it managed to avoid major drought and desiccation losses and most of the flood and scour losses, dampen that variance in fry to spawner values, and that's enhanced the I guess the availability of fish for reintroduction efforts within the Okanagan Basin because they get the brood from the Soyuz returns, and then also the Yakama River, and ultimately we hope the Upper Columbia. So I don't know, do I have time for one conclusion slide, or am I done?
Yeah, just to keep on track of it. I guess, well, go for it, I guess. Be quick. Oh.
What's necessary for the population persistence? We're going to lose the Soyuz at some point. We need to focus on Skaha and Okanagan lakes. We need to create habitat there. We need to give them robust, strong, consistent escapement so they can develop the diversity they need to capitalize on new spawning opportunities like shoreline spawning, like the kokanee in Okanagan Lake. We need to manage our fisheries to account for the escapement needed for the Okanagan, and you can see the rest. Yeah. Thank you, Ryan.
All right. We will have that Q&A at the end, so. All right, our last talk of this portion of this session is Dr. Michel of the Upper Columbia United Tribes, and he's going to continue to take us upriver with his talk on the restoration of native fish populations to their historic habitat throughout the Upper Columbia. I was going to say, well, they're doing a little technical issue, a little shameless plug real quick. Here in the Upper Columbia Salmon Recovery Board, we are hiring for a job, the Salmon Habitat Recovery Project Manager. So if you know anybody who might be interested in a position, go check out our website. So I was told to use the time to do a little shameless plug. So there you go.
Afternoon. We after lunch or pretty quiet out there? I'd like to thank the board for providing us this opportunity to visit with you today. Again, my name's Michel. I'm a citizen of the Lakes and Cobble Bands of the Confederated Tribes of the Cobble Reservation. I've been the executive director for UCUT now for 16 years. We take a proactive, collaborative, and science-based approach to promoting fish, water, wildlife, diverse habitat, and tribal culture in the Northwest. We work for nearly 15,000 tribal citizens. Together we manage and influence nearly 2 million acres of reservation land and influence at least 14 million acres of Aboriginal territories, 500 miles of waterways, 40 interior lakes, and 30 dams and reservoirs. The organization was formed in 1982. We came together to ensure a healthy future for the traditional territories and lands of our ancestors. As sovereign nations, we are charged with the protection and enhancement of our natural resources. Our mission statement, I think, is unique in that it calls out who we are and what we need to do as people to bring back what was taken from us with the development of the Columbia River Hydro System and other things around the region that have had negative impacts on our fish and wildlife. Our mission statement is to unite the Upper Columbia River tribes for the protection, preservation, and enhancement of treaty, executive order rights, sovereignty, culture, fish, water, wildlife, habitat, and other interests and issues of common and so I want to, I'm sorry for the slow reaction to some of the words and some of the things we've got going on. I've got a video here. It's about 10 minutes. It talks and shows the absorber of those tissues and how the fish have moved in and what they do. So economically, the Columbia River Basin holds immense natural capital value. Ecosystem services are estimated to provide a value of $190 billion annually. This value exceeds and dwarfs all other economic values. For comparison, hydropower provides a value of $3.3 billion. So when you start to look at those barriers, those foundations, that money is a lot more if we look about this in a whole picture. What those things do as it relates to the ecosystem are more valuable, have more to offer than what's available in just looking at, for example, the dams and what they provide. So with that, we'll show the video. It lasts about 10 minutes, and it has the 5 UCUT tribes and what they go through and how they use those things and how it impacts and benefits us as we go forward.
Fish were the main staple, the main diet for our people for thousands of years. But it also was an opportunity for us to engage with other tribes in the area as well. And it really wasn't until the dams were put in place that blocked the runs from coming up into our location that really made it challenging for us.
The history of the Cooney tribe of Idaho, our oral history tells us we have a covenant with the Creator. It says, as long as you take care of this land will in turn take care of you. And of course, we like the benefits that the dams bring, but the dam does bring issues. We realize the sturgeon were in decline.
The impacts of hydropower aren't just on the river. It's in the margins, it's in the mountains, it's on the people. Both salmon and resident fish are the fundamental protein resource for the Kalispel people. And so what happens when that's gone, right? You start to have secondary and tertiary impacts.
With the construction of Grand Coulee Dam, it changed everything about the river. It blocked the salmon, it changed our way of life.
The Colville tribes, we are river people, and losing those salmon for the amount of time that we have has made us incomplete.
The way we eat, the way we gather, the way we practice culture and ceremonies. So it changed. It was life-changing to lose the salmon.
You hear some of the stories about when that happened. The tribes weren't informed, weren't let know. When the salmon didn't show up that first year, thought they'd done something wrong, thought that they were being punished for something.
We are the people of the Upper Columbia. Our people could not imagine life at one time without the salmon.
The Upper Columbia United Tribes is a 5-tribe organization. We're in the blocked area above Grand Coulee, and it's the Kalispel tribe, the Coeur d'Alene tribe, Kootenai tribe of Idaho, the Spokane tribe, and the Colville tribe. And most of what we do is working on mitigation issues around the impacts of the Columbia River hydro system.
UCUT's kind of been that central location then where some of our brightest, best and brightest have come together to figure out, okay, if we're going to reintroduce salmon, what is that going to look like? How is that going to be?
Somewhere along the line, we realized that if we can get rid of the stuff that we don't agree on and just focus on the things that we do agree on, we can get a lot of things done.
You look around the basin in the Upper Columbia and you see the tribes at the forefront of resource restoration, hydropower mitigation, working really strongly with our collaborating agencies.
Idaho Fish and Game, Montana Fish, Wildlife and Parks, BC Ministries, BC Hydro, Bonneville Power Administration, Army Corps of Engineers, United States Fish and Wildlife Service, Idaho Fish and Game, so many different entities, alphabet soup of, at these agencies.
And at first it was a fight, and then it became a collaboration. And now we have a web of partners that are helping us achieve the vision that our tribe set forth decades ago.
I think for a long time people were kind of thinking that it was a choice between salmon or energy. Everybody's realizing, especially with reintroduction, that you can have both.
For the benefit of all is a very important part of who we are as a tribal people. The tribes aren't going to be the only ones that benefit from fish passage.
I know in this room, the room is set up ultimately for us to be able to rear young fish, young salmon in particular, so that we can be able to ultimately pit tag them and track them as they head down to the system all the way out to the ocean and hopefully come back. Last year we had our very first adult return from the ocean. That was really exciting, and we had a big ceremony when we put them back in the waters in the blocked area.
Those ceremonial releases have been just beautiful. We had one of our elders actually release a salmon at Kettle Falls, and he was there for the ceremony of tears as a child. And so to see salmon returning back to the falls was actually a very beautiful, very moving moment.
We have a facility here at Box Canyon that has an upstream passage facility that's in operation. We're working with the Corps of Engineers and the Bonneville Power Administration for final design and construction of upstream fish passage facilities at Albany Falls Dam. The goal's restoration of a trust resource like bull trout for the Kalispel tribe.
What we have in these tanks behind us are Cooney River white sturgeon. We have the brood stock. We're in the middle of spawning right now, so we have a lot of the big mamas in the tanks right now. The tribe has been doing extensive habitat rehabilitation work. And so part of that is fixing side channels that have been deteriorated and making them viable again. And so that allows for other fish to go in there and rear and raise and have a place to hide and, live.
Who we do this for is my great-granddaughter.
We want our children, our grandchildren to be able to enjoy some of those same societal factors that we saw thousands of years ago.
We've had our nose at the grindstone for work, and now we're up the mountain quite a ways. We can finally take a look and see how far we've come. And we've come so far. It's such a wonderful time to be able to create life.
And for the Spokane tribe, we believe that when the salmon return, that will bring healing to our people. Hopefully someday we're able to see 20,000 fish come home or 50,000.
To be able to see those salmon rippling in the water when we turn them loose for those cultural and educational releases, to see them swimming in those waters after 80 years, it's just an amazing feeling. And to know that it can happen, and to know that you was a part of it. This is an opportunity again for us to heal, for us to fill that void in some of those folks who I've talked to since those releases, and that reconnection has changed their life, who they are, and has helped bring them back, has helped bring them out of that. So it's more than, it's more than just a salmon. It's who we are, and who we need to continue to be, and we will. But it will be better when the salmon are home.
Our people could not imagine life at one time without the salmon. But now we are seeing them again. And that means we gotta pray harder, together, because they're listening to us.
So I'm asking DR to stay up here, as well as want to invite Josh, Jose, Jeff, and Tom to come back up for our Q&A time. So, and just like we did earlier, we'll have questions to the standing mic, and also we'll have a mic runner as well. So.
My name's Ginger Wireman. I'm the new water quality lead for RIA's 46 through 50, so NTIAT all up here for the Department of Ecology. I've met a few people in the room, but I actually came from a communications background. and in particular have had training in community-based social marketing. And I just really appreciated what David was sharing with us. And I also have done a lot of training in climate education. And one of the things that is showing up really concerning is that the children are being told that everything is doomed and they're not being given solutions. So I think I would really want to encourage everybody to look back at David's research and see where you can incorporate it into your work. And then DR, I'm not crying after that, not at all. That was lovely, and thank you for sharing it with us. And I think it shows us the importance of stories in helping us do our work and share the importance of our work.
Thanks for all your talks. I got some questions, I suppose. Jose, what are some potential funding sources that you think might exist for bull trout? Where are they now, and where could they be in the future?
That's a good question. As a federal employee, my options are somewhat limited, but I can help you all by being matching funding on a lot of different grants. There are others in the room that I would probably toss that question to. There are definitely, we've applied for surfboard grants before with mixed success. I know everyone in the room is probably familiar with some of those projects. There are sources out there that are competitive for all species, which bull trout projects in the areas have, again, mixed success being funded with. But yeah, I'd say get back to me on that one specifically, because I've got some other people that would be able to spit out the list. I can tell you all the things I'm not eligible for. But if anyone out there, actually, seriously, I'll give a plug. If anyone out there does have a project that they're considering and would like to talk to us about involvement and, potential matching, we can at least talk, and, maybe we can point you to someone who can help if we can't. So.
Keep going if nobody else has any questions. For Jeff, what happened in 2020 with the returns? You showed that chart and things went like tenfold for the last 3 years.
I'm trying to think 2020. I don't think there was anything specific. I would have to think about what rearing conditions looked like a couple years before that, but off the top of my head, I don't have an explanation for 2020 in terms of returns.
Well, just like the last 2020 to present, the last 3 years all had like an order of magnitude more.
Yeah. I think, a lot of it, I think, is just, it's taken us a little bit of time to really, fine tune some of our collection and spawning methods and, some of the fish husbandry part of it. And also, like I said, it's just, years and years and years of, like I said, fine-tuning, the hatchery part, as well as just, beefing up that brood stock and, increasing local adaptation. So why 2020 was the beginning of the, that big increase, it's hard to say. But I don't know. Usually when you don't know the answer to those things, you just say ocean conditions, us freshwater people, so I'll say ocean conditions.
I think this is my favorite session so far because we're talking about success, and we don't hear much about that, so kudos to you all. And my question is mainly for Tom and Jeff. DR, this might be something that accounts to you at some point, but probably not now. But we've seen some other talks earlier where we talked about density-dependent effects. My question is, I know that we want to revel in our successes, but what are the trade-offs with the successes that you're having in coho and in sockeye? Are there any downsides to any of the other stocks or limitations that you guys can see in the future?
downsides to success with sockeye? Well, in terms of habitat improvements and restoration of access, that seems like that's actually going to be beneficial to other stocks such as Chinook and steelhead and coho. I'm not thinking of any downsides. You have any? Did you have something in mind? Is this a trick question? Or, yeah. Scale, yeah. So the nice thing about sockeye is that they're lake rearing, and the other species don't tend to be. And we do get coho rearing in lakes some, and once in a while Chinook, but sockeye have their unique habitat, and they're spawning. I guess the Chinook are spawning on top of them, some, but I don't see them. And I guess the fall Chinook will be spawning about the same time, or summer falls. So I'm not seeing any downsides. Lots of carcasses.
Yeah, so in terms of impacts to other species, coho are a little bit unique. They're fall spawners. We have Chinook in the system, but spawning timing is a little bit different, and they're using different size gravel for the most part. Obviously, bull trout, ESA listed species are always a concern, but we've done predation estimates and we've done some other stuff where we don't see a ton of overlap in coho and bull trout. And I guess, one of the issues, like some of those years we've had those big runs, a lot of those fish are just going back to the icicle, and they're basically just superimposing on themselves. So having a lot of coho in the Wenatchee Basin is actually somewhat of an impediment to coho, which is why we're trying to, get these fish to, spawn and establish in some of these further upstream reaches, so they're not all just spawning on top of each other.
I got a question for Josh. With the bull trout movement in the lower and middle zones, and seeing fish move from those areas that were either tagged down there or detected down there and moving into the tributaries, you were talking about that movement for cold water. Were those juvenile fish that you were also detecting moving back out, or were those fish moving into cold water and to spawn? So did you see movement of juveniles back and forth between the kind of main stem habitats and the cold water? And then kind of a second part of that whole thing, within that whole study, were you able to glean any kind of sex, male-female differences between the movement patterns?
Yeah, good questions. So for most of the fish tagged in the lower and middle areas, most of those fish that we got detection histories off of were adults. Most of those have been fish sort of detected at sites that we wouldn't expect to get many juveniles. So they were, adult salmon collection facilities that also just happened to get adult bull trout. So we don't have a lot of juvenile data for the lower river. Almost all the small juvenile bull trout information comes from the Chihuahua, from either our backpack electrofishing or the small traps. So most of that movement was specifically just for adults, for the most part. And then your second part of the question was sex, right? If there was sort of male-female stuff. We don't have any real good gauge for doing sex determinations at this point. We have run ultrasounds on bull trout at like Tumwater Dam to try and get at sort of looking at the gonads, but that has its own host of issues to sort of the false positives or negatives there. There is we haven't been able to really tell sex is the simple part, but there are some opportunities that might be coming forth very soon on using genetics to distinguish sex. There's a new sort of panel or whatever that can distinguish that, and so all the genetics that we have taken or any genetics that we take in the future, we should be able to run that and not only get sort of tributary assignment, but also a sex of the fish, which would be really cool and useful data to get at. But at this point, we don't have a real good gauge of sex. Yeah, thank you.
I'd just like to touch a little on some of the success and things we've done in the Upper Columbia as it relates to releasing cultural and educational releases above Chief Joe and Grand Coulee, and the success and some of the pushback we got early on that those fish after 80 years wouldn't know where to go or how to utilize that habitat, and that most of them would just turn and go back downriver because they hadn't been above those two facilities in 80 to 100 years. And as we started those cultural and education releases, that was far from the truth. Those salmon, for the most part, got above those dams and went upriver, looking for that pass, looking for that way home. And home to us is the headwaters, and all of that. We get hung up on trying to find habitat and cure our problems by doing what we've been doing for the last 50, 60, 70, 80 years. And the development of Chief Joe, and that the river doesn't stop at the base of Chief Joe, that there's miles and miles of good, clean habitat above those two facilities, getting them fish back up into Canada where they belong, home to the estuary, is an important part of what we're doing. And for the most part, it's been very successful. They've seen the fish up in Canada at the Keenly side. Actually, a few of them have made it through the locks there when they're letting boats and things through. So I just wanted to share that with you, that those salmon are just waiting for us. They're looking for that opportunity to go home. With the development of the system 80 to 100 years ago, we eliminated that, but those fish haven't forgot. And I think that's the important part of where we're at. And as we go through these conferences, those fish remember where they came from. And it's our responsibility for those future generations. You mentioned my great-granddaughter in the video, and I've got several grandkids, and that's who we do this work for. It's not for me. It's not for us today. It's for those future generations, those people we haven't met yet. So as we go forward, keep that in mind. There's an opportunity to correct a huge historic wrong that will benefit all of us, culturally, spiritually, and economically. And I think that's something that we need to touch on a little bit too, is the economics of fish passage and the opportunities provided in that. It's not going to cost us. It's going to provide us a lot of economic opportunities. So as folks say, we can't afford to do it. I say we cannot afford to do it right now. We're looking at power production, flood risk management, and the incorporation of ecosystem back into the Columbia River Treaty. And we can use all the support we can get, your congressionals, your locals. Talk to those folks and tell them this is important and we need to do it. So thank you. Thank you.
I think with that, let's go ahead and thank all our speakers for this part of the session. So this next 30 minutes is a break, but it's also the poster session. So I checked out the posters during lunch. There's some great posters out there, just kind of through the lobby there. So go check out the posters, and we'll see you back here in 30 minutes at 3:30.
I know.
Good. I am presenting the next block. The next block? Yep. The last block. Yeah. What's your name? Lance Keller. You have a 415 slot. Yep, no worries. There should just be nothing else in the room besides the PowerPoint presentation and PDF copy. All in. Okay. It, yeah. Yeah, I need to still put in to bring that presentation. I need to be able to switch this on this. Okay. So in the future, that I can have it, so that I can bring it back. If someone has notes, they can see it. Yeah. Yeah, but no, that's sweet. I saw them on the way down. I'm like, oh, is that far enough? That's a nice, yeah, that's nice and clear. So very nice setup. Thank you.
Bye.